Hi, my name is Christian Freeman, and I study lipidomics in the Heinz lab here at UGA. Lipids are molecules that consist of long carbon chains and are often insoluble in water. In all living things, a certain class of lipids known as phospholipids make up majority of cell membranes. Phospholipids contain two fatty acid chains bound to glycerol with a phosphate group. Phospholipids can vary in different head group classes, and the most abundant found in bacteria are phosphatidylglycerols, or PGs. Phospholipids have uniquely shaped fatty acid chains while having the same number of atoms and atomic weight. These are called isomers. My research focuses on separating isomers that differ in branched or straight chain fatty acids found in bacteria by using reverse phase liquid chromatography ion mobility mass spectrometry, or RPLC IMMS for short. RPLC is a chromatography method that can separate molecules based on their solubility in different solvents. Through a custom gradient of solvent percentages, I can separate phospholipid isomers enough to view the differences in composition between bacteria strains that differ in growing conditions or mutation. Each sample is passed into an ion mobility mass spectrometer, where each phospholipid isomer can be identified and quantified by mass and intensity. The ion mobility also assists in filtering other ion signals, assuring we look at the right lipids. I use this method I developed to assess the differences between branched and straight chain fatty acid isomers in Staphylococcus aureus. Phospholipids containing branched fatty acids are used to increase membrane fluidity. Membrane fluidity and polarity plays a large role in bacteria functions or antibiotic susceptibility. Currently, I am looking at the differences between wild type and a daptomycin resistant strain that contains a mutation in PGSA and NPRF. Two genes responsible for the synthesis of PG is positively charged derivative lyso-PG and lyso-PG relocation. By looking at these two most abundant lipids, I can look for a correlation between daptomycin susceptibility and membrane fluidity or polarity. As of now, we do see a correlation between PG and lyso-PG shifts that support our hypothesis about increased membrane fluidity and polarity, reducing antibiotic susceptibility. So, what's the end goal? Lipidomics is an underdeveloped field compared to proteomics and metabolomics. Advances in this project would spread the importance of it by drawing further connections between cell survival through membrane stability. Methicillin resistant Staphylococcus aureus is also one of the leading causes of bacteria infection around the world, and bacteria are constantly developing resistance to new treatments every day. Verifying this connection would allow us to turn the timetable and increase susceptibility in older antibiotics, changing the playing field that's proposed significant challenges today. In conclusion, I would like to thank the ARCS Foundation for their support of the fighting against antibiotic resistance. The assistance of this scholarship will be used to fund national organization membership, conference registration, and personal skill classes to support the development and communication of my research to others, including at the American Society of Mass Spectrometry meeting this fall.